Welcome to the last video of this course. I hope you've enjoyed learning about the cellular mechanisms of brain function. Our goal in this course was to obtain a causal and mechanistic understanding of how the mammalian brain works. Because the brain is made of cells, in any mechanistic explanation of brain function, it's essential to think about the activity of individual neurons and their synaptic interactions. The brain is a very large, synaptically connected neuronal network, and the complexity is bewildering. We're only at the beginning of a great adventure, and clearly we still have much to learn through further neuroscience research. However, as we've seen, modern neuroscience is beginning to provide answers about the causal mechanisms underlying simple behaviors, at least in mice. And much of what we learn from studying mice will have direct implications for understanding human brain function and dysfunction, and will likely give rise to novel therapies for various brain disorders. This course has focused on the biophysics of brain function, seeking to explain the millisecond by millisecond processing of sensory-motor information by neurons, synapses, and neuronal circuits during behavior. We've tried to bridge different spatio-temporal scales of analysis of the mammalian brain, from nanometer-size ion channels transitioning between closed and open states on the microsecond timescale, ranging to the investigation of intact sensory-motor neuronal circuits spanning a centimeter or so in the mouse brain and operating on behaviorally relevant timescales from milliseconds to days during the learning of behavioral tasks. Remarkable technological advances are driving the discovery of new aspects of brain function through measurements and perturbations with increasing specificity in behaving mice. It now seems quite likely that in the next 50 years or so, we'll have a very good understanding of how the mouse brain works. Obviously, in a short course of 7 weeks, it's only possible to cover some selected topics. I hope this course has provided you with a good foundation, allowing you to think further about how the brain works. And I hope this course has whet your appetite to learn more. Here we've taken a biophysical, bottom-up approach to studying the mammalian brain. But there are many other important perspectives, many of which we barely touched upon in this course. For example, developmental studies and comparative studies across species will help us understand the logic of how the nervous system's put together, giving important information about general organizing principles. We only briefly mentioned some aspects of molecular biology and genetics, and there's much more to learn about molecular neuroscience, both in terms of providing sophisticated tools for studying the functioning of the mammalian brain, but also in terms of modeling human genetic brain disorders in mice, with the hope of better understanding brain diseases in order to provide rational treatments. Finally, as we obtain increasingly detailed information about how the brain works, it'll become more and more important to quantitatively test these hypotheses through computational modeling, and it's therefore important for the modern neuroscientist to be intimately familiar with mathematical modeling methods. So I also encourage you to spend time studying computational and theoretical neuroscience. With these last thoughts in mind, I'd like to thank you for your participation in this course and to wish you all the best for the future.